Aracil-Gisbert, and I'm going to present today this work titled "The Microbiome and Resistome in the Intensive Care Unit Patient Care Environment, a longitudinal multiomic study using pooled fecal sampling." This work is based on that the risk of selecting, acquiring, and spreading opportunistic antibiotic-resistant pathogens within the hospitals is really high in the intensive care units. So in this work, we evaluate the contribution of the patient microbiome and the antibiotic resistome to the transmission of antibiotic-resistant genes within the intensive care unit. To do that, we perform a longitudinal study in the intensive care unit of our hospital in which we recovered samples, fecal samples, from patients during a year. These samples were divided into two groups: the newly admitted patients group, or group 0, that were samples recovered during the first 5 days at admission, and the long-term patients, or group 1, that were successive samples until the discharge of the patients. Those samples, we selected one sample per patient per week and per group, and we extracted the DNA and performed the pooling analysis, in which samples from the newly admitted patients were pooled into pooled samples P0, and for long-term patients, the samples P1. Those pooling samples were submitted to two metagenomic approaches. On the one hand, to analyze the microbiota composition using 60-cell RNA sequencing, and on the other hand, the antibiotic resistome using RESCAP, a targeted metagenomics approach developed on our lab. Looking into the results, if we focus first on the microbiome section, we can see here in the panel A the alpha diversity of those pooled sample groups, in which we can see that the newly admitted patients had higher diversity and richness than the long-term patients. Also, we analyzed the beta diversity that you can see in this ordination plot from panel B, in which the taxa distribution of those groups was significantly different, being long-term patients more heterogeneously distributed within this taxa distribution, and also this group was enriched in gram-positive bacteria, such mostly Enterococcus and Staphylococcus, as you can see here in this left analysis in panel C. In panel D, you can see the composition of these different groups, in which the composition was significantly different due to the recovery of the less number of taxa distribution in the long-term patients. Looking into the antibiotic resistome section, we can see here in the panel A also the alpha diversity of the number of genes that we analyze, in which the newly admitted patients had higher diversity than the long-term patients. That is consistent with the results of the microbiota. In panel B, we can see the composition of this number of genes that are divided into the different classes of antibiotics, in which the composition is different, having the newly admitted patients more number of genes than the long-term patients. Also, we focus on the clinically relevant antibiotic-resistant genes for our hospital, for some genes that are involved in outbreaks within the ICU. And we can see here in this panel C, in which we can see in the x-axis the week of sampling with the number of genes that we analyze. In here, we recovered genes that were conferred resistance to third-generation cephalosporins and carbapenems, mainly extended-spectrum beta-lactamases and carbapenemase genes in Enterobacteriales and Pseudomonas, such as KPC, OXA48, BIM, or CTXM. Also, we found genes conferring resistance to beta-lactams in Staphylococci with the representation of the MEK-A gene, and genes conferring resistance to glycopeptides in Enterococci with the representation of BAN genes. Here, in conclusion, during this work, we can see here that the extended ICU stays significantly modify the gut microbiome composition of the hospitalized patients, particularly reducing the bacterial diversity and the gene content. Also, that the dynamics and clinically relevant antibiotic-resistant genes, mainly ESBLs and carbapenemases, during the outbreak periods are predominant. And the persistent detection of other genes for gram-positive bacteria, such as MEK and ban alleles may reflect distinct selection and transmission pathways driving the spread of bacterial species in the ICU. Thanks for attending and see you in the venue.